Thank you, Jen, for the introduction. I also want to thank the organizer for this wonderful opportunity for me to learn this many interesting topics today. So I'm going to talk about the brain again. So I want to start by saying by asking questions why understanding brain is so difficult. I think at least we can think of several very important hallmarks of the brain, which one of them is extraordinary structure and the functional diversity of the brain. For example, cell types can be defined by many criteria. So we can define them by morphology, electrophysiological properties, molecular markers, and so forth. And also, it has been mentioned many times in this symposium already. Brain is organized at multiple spatial scales. The brain networks are composed by many regions as in a millimeter or centimeter scale. And neurons, the cell body is about 10 micron, but their dendron synapses can go down to 100 nanometer scale. So this is really a wide range of spatial scales you have to cover to understand how brain works. And we long ago go back to Neocortical Hall. We understand that the brain cells are really they have very distinct morphology in different shapes. When the cells are located in different regions and in different locations for given region. So cell type organization is really fundamental for understanding brain functions and also for brain disorders. So now it's really an exciting era in understanding all these brain cells. For example, the recent BICCN consortium has generated a really nice comprehensive multimodal cell census and atlas of, particularly in primary motor cortex. So we really, with the new exciting technology emerging, we have a very good starting point, I would say, to really understand the cell types and how they located in the brain. So the spatial transcriptome, there's no need to introduce, has been mentioned many times. It's very important by Friesen and Underberg. Now we can use this to really understand the cell type organization of the spatial organization of the brain. So I'm going to focus on the macaque brain. And I think monkey is a very good animal model because it's evolutionary, it's very close to human. Mouse, for example, there are about 70 to 80 million years separation from humans, and macaque is the closest animal models that we can perform a really comprehensive study on them, which has 35 million years separation. And in terms of gross morphology of the brain, you can see that macaque brain is much closer to human brain compared to mouse. And also, macaque has a very complex social behavior, and also like the two use and social observation is very difficult to observe or study other species. So I think macaque brain is really a very nice animal model for us to understand how our complex behavior emerge from a neuropsychic. But we do need a very good understanding of cell types of non-macaque brain and also how those different cell types are located. That's where Stereo-seq from BGI comes in. And I think I don't need too much introduction, but when we got to know this technique 1 year ago, we were very sure to know that this exciting technology has the very large coverage because the size is very large, and also the very high spatial resolution because the different DNA nanoball is very small. It's like you have 500 nanometer resolution. Is still have the space to improve. We can, if you want to go to like nanometer level, I think we need to also include expansion technique and so forth. But now we have a very good technique to study the spatial organization of cell types in situ in macaque brain. So we assemble a large team work. So we section the macaque brain at the coronal section. So what we show here is the protocol we use. So we develop a technique to section the frozen brain at 10 micron thickness to perform this Stereo-seq analysis. They will also collect the neighboring slices to perform single nucleus-RNA-seq as well as the immunohistochemistry staining for anatomical pixelation. So in total, we perform this experiment 3 macaques, and in one of them we have very nice coverage. We have total 120 something slices from one monkey, and so from the then you can get a very nice separation of cell types based on the SNO RNA-seq, and then using this cell typing, you can register your cells with on this Stereo-seq data to get the spatial organization of the different cell types. So this is the example images of raw mRNA. So this is enlarged tissue image. You can see that there are cell shape type of aggregates of mRNAs on this chip. So indicating those really you have this single cell level resolution of cells. We use autocorrelation based method to circulate the cells marked by red here. And for each cell, we get about 500 to 600 genes captured by this technique. So as I mentioned, we use the SNO RNA-seq technique to first identify different cell types from the macaque brain. So this is the data from cortex based on about 500,000 cells throughout the entire macaque brain. We identified about 200 different types of cells, and then we registered the cell types back to the Stereo-seq spatial transcriptome data. You can see that each cell on the y-axis, different cells. There are very high, typically only one cell types, a very high correlation with this, so indicating a good registration. So we first look at the gross spatial location of different cell types. You can see that very remarkably there are many cell types has a very interesting gradient from anterior to posterior level. So there's a drastic difference for many cell types in the macaque brain. So this is a global 3D cell atlas for the entire macaque brain. It's about 20 sections of the macaque brain. So you can see from the different color indicating for different cell types. So this is really a comprehensive. I think it's really nice to see this image. For an anatomist, you can clearly see boundaries of different regions. And potentially, this data could be used for fine pixelation of the brain and other purpose. So I'm going to show you 2 examples. So this is elongated brain showing the different several example cell types have very different location from anterior-posterior axis. And this is several examples cell types for the subcortical regions. You can see also clearly they have different locations. So you can dump into data to look at many hallmarks features of the brain. One hallmark is layer specificity of the brain. So cortex, neocortex can be separated into 6 layers. And if you look at the Stereo-seq data of the macaque brain, you can see a very clear layer specificity of the different genes. This is showing several examples. You can see for different regions, there are those different genes. They have a very distinct expression pattern across different lambda distribution, revealing the layer specificity of those genes. And also, as I mentioned earlier, we can also see many region-specific gene expression profile. For example, those different. So what we show here is a plot from 6 different genes. They have different localization throughout the. So this is dorsal. This is the ventral part of the neocortex. We can also use the registered cell types, and we found several cell types have regional specificity. For example, this cell types has a enriched distribution in area 3, primary sensory cortex in here, and this cell types has enriched localization in primary motor cortex, and you can also see increased expression level of example marker genes. So there's a region-specific distribution of cell types and gene expression discovered by the Stereo-seq. So those are the neocortex. We also found similar region-specific gene expression and cell type distribution in subcortical regions. This is example image beautifully show the expression levels of different genes in like colostrum, putamen, thalamus, and so forth. So it's very clear that many genes and many cell types show distinct distribution. And this is an example from thalamus. You can use autocorrelation method to find those different cells, and actually the different regions exhibit distinct expression of certain genes and different cell types. So clearly you can find region-specific gene expression in subcortical region. So this is a very, I think, important for many research, because you can then delve into the data to see your interested region and what genes are highly expressed, therefore, and what cell types there are. So you can do specific manipulations and recordings their neural activity during certain behaviors. So it's very, I think, very important for future functional study. I'm going to show you 2 example regions that we did a little bit more in-depth analysis. One is hypothalamus, has been shown earlier in the symposium. It's very important for many innate behaviors and controlling homeostatic function of the body. And so we look at the entire hypothalamus that's located here, and you can cluster cells into different types, excitatory and inhibitory neurons. Very interestingly, different cells, there are marker genes. The top marker genes typically have neuropeptides in there, right? So I think it's very important. So that this is different neuropeptides. This is consistent with literature, but it's nice to see this on this global scale. And for these different cell types, they really have very distinct localization throughout the different subnuclei of the hypothalamus. We can just very clearly see these different cell types indicated by a different color. They are located in different subregions of hypothalamus. And also the cell types and the gene expression level are very closely related to each other. So I think this is a very nice data set where we have a team that we are actively examining this hypothalamus in detail. What's the molecular and the cellular landscape of this transcriptome. Another example region is colostrum. Colostrum is a very interesting region. It's a very thin, sheet-like structure. It's located very deep inside the brain, and has been implicated in many brain important functions. For example, consciousness, proposed by Francis Crick and Cliff Koch. but this region has really understudied, I would say, in terms of cell typing and their spatial localization. So we examined the spatial transcriptome on classroom and found several cell types. They have very distinct localization. So at least you can see that some cells show this media preference, and other cell types show this preferential localization on the edge of the classroom. So there's clear spatial localization specificity for those two cell types. So I'm going to summarize. So we found by using this new Stereo-seq technology from collaboration with BGI, we found distinct spatially resolved cell type composition across neocortex and subcortical regions at whole brain level from our cat brain. And we found distinct layer region-specific genes present profiles for cortex and subcortical regions. And I didn't talk about for the interest of time, we can also use this spatial transcriptome analysis to postulate the brain in a more in precise fashion, and also in depth analyze the transcriptome landscape for the hypothalamus in the classroom. I didn't show data, but we found the whole brain topography and the coordinated expression pattern for important genes, for example, neurotransmitter and neuromodulator receptors and transporters. Those are the very important target for pharmaceutical companies for this brain diseases. So now the manuscript has been uploaded to our archive. You can go into the manuscript if you're interested in the detail. So now we are delve into the more analysis, because it hasn't been mentioned in this conference. The really data analysis is the major challenge. One of the major challenges for this field, I think, we spend a lot of time trying to dig into the data and find the principles for our curiosity and to understand the brain. So I think there's a lot of things we can do. For example, for disease brain and for aging and for development, it'll be very interesting to compare mouse, macaque, and human, and also will be important to study connectome. We can use retrograde tracing technique coupled with spatial transcriptome to understand how the different types of brain cells, different regions are connected with each other. So with that, I would like to thank the team who really did the work, and it's really a huge team effort. So we have more than 10 PIs from our institute, and Zhiyong Liu, Zhiming Shen, and others. They are heading the experimental team, and there's Yidi Sun, Zhifeng Liang, and Wei Wu. They are heading the analysis team, and Yangang, Mu Bing, and I are leading our team effort. And from BGI Research, I would really want to thank Dr. Wang Jian, Xu Xun, Liu Longqi, Chen Ao, and Yu Xiang for their wonderful work. It's really a joy to work together. We really work no stop for more than a year, and this is Chinese. I think the second day of Chinese New Year. So we are discussing how to prepare, how to design the project. So with that, I would like to thank you all, and I wish STOC great success. Thank you.